7.1.9 is asking us to look at an experiment by these two scientists named Hershey and Chase. And their experiment finally gave evidence that DNA was the genetic material. Before this, they didn't know. So if you remember, DNA is this double helix, but also in eukaryotes, it's associated with proteins. Now, there's something called histones that wrap the DNA up into like very manageable pieces. So a histone, which we will talk about in 7.2, histone is made of 8 small circular pieces of protein like this, and one more piece of protein that kind of gives it like stabilizing on the side. And then what happens is the DNA, which I'm going to represent in blue, wraps around, you can visually wrap around in here, wraps around these proteins. This structure is called a nucleosome. So they knew back then they knew back then that the genetic material, your chromosome, was made of DNA, but also it was made of protein. So the mystery out there was, which one has the genetic code on it? Is it the DNA or is it the protein? So Hershey and Chase came out with a pretty cool idea about how to figure this out. So they did 2 separate experiments using something called a bacteriophage. So bacteriophages are these really weird viruses, and they specifically target bacteria. And how viruses work is they can't replicate on their own. They need to inject their genetic material into a host, just like when you get a cold or a flu. They need to inject their genetic material into a host, and then the bacteria are made using the protein assemblies by reading your gene and eventually turning that into a protein. So that's how viruses attack. They need to inject their DNA into a host. And in this situation, the host is a bacteria. So that's what this circle is supposed to represent. Okay, so let's talk about how they designed their experiment. So I'm not exactly sure what order they did it in, but let's just pretend it's this order. So what they did is they made viruses only replicate, or the only source of protein that these viruses could use is sulfur 35. So this is a radioactive isotope. Now, sulfur, if you remember, is a component of a lot of proteins. A lot of amino acids. So we talked about cysteine forming those sulfur bridges. Oh, I spelled amino wrong. Cysteine is quite common in protein structure. So if we made sulfur radioactive, we could follow it. So sulfur's in the amino acids, which basically means proteins. So they made these viruses protein coat. So the outside, I'm going to use a different color here. The outside of the virus right here is all made of protein, and the DNA is in the inside. So these radioactive viruses with their radioactive protein coats injected DNA into the host cell. Eventually that DNA was turned into new viruses, but not with radioactive protein, because the protein was stuck on the outside. So what they did next was they put this solution, because this was all happening in a solution, they put this in a test tube. They spun it at a super high speed in a centrifuge, and everything that was heavier ended up at the bottom, and the lighter stuff ended up at the top. The viruses, which are smaller than this bacteria, they were kind of like floating around in the top of this solution. And then the heavier bacteria were found down here at the bottom. And they could identify, right, that the lighter radioactive portion was floating in this top part and not down here. This part of the test tube is called the supernatant. I don't know why they call it that, but supernatant is this solution of the where the viruses are. Supernatant is a real word, a really weird word, I know. So in this situation, they were like, okay, no DNA down here in the bacteria, but let's just make sure. So then they redid the experiment, and this time they used phosphorus 32. So this is a radioactive form of phosphorus. And phosphorus, if you remember, oh yeah, DNA nucleotide. So nucleotides are made of a nitrogenous base, a sugar, and of course this phosphorus up here. So I'm going to erase this in this situation. So let's recreate the same experiment, but this time our DNA is radioactive. So I'm going to put that in yellow down here to signal the radioactive part. And viruses inject their DNA into their host, as we mentioned earlier. So inside here, the bacteria was filled with radioactive phosphorus. So when they centrifuged this solution again, this time the radioactive part of the solution was down at the bottom, indicating where our bacteria were. And the non-radioactive part was up here floating around in the lighter virus. This part of the experiment, they gave it a special name. So the liquid part is a supernatant, and this down here where you find the bacteria is called the pellet. So that's how these guys did it, using two different types of radioactive isotopes. Sulfur, we could identify where the proteins were. And when we made the sulfur radioactive, we found all the proteins were in the supernatant, connected still to these viruses. And then when we made the phosphorus radioactive, it was injected into the bacteria. And then all the radioactive phosphorus was stuck down here in the heavier pellet made of bacteria.